Hello everyone, my name is Orsolya and I am going to talk about my PhD project where I investigate neuroimaging and neurophysiological markers of schizophrenia. I am a psychologist and I work at the Department of Psychiatry and Psychotherapy at Semmelweis University. My supervisor is Gábor Csukli, and my vision is to understand the underlying neurobiological mechanisms of schizophrenia, and in order to reach that vision, my mission is to study potential neurophysiological and neuroimaging markers of schizophrenia with tools such as transcranial magnetic stimulation or MRI. Here you can see my specific goals to reach this mission and vision, and I am going to talk about them in more detail. In my first project, I am investigating motor cortical transcranial magnetic stimulation paradigms as potential biomarkers of schizophrenia. Schizophrenia is a very severe psychiatric disorder. It has a prevalence around 1%, which has shown a significant increase in the last couple of decades. Schizophrenia can be described with positive, negative, and disorganized symptoms, which already show you how heterogeneous this disorder is. This kind of heterogeneity is not very well explained by current diagnostics because we don't fully understand the neurobiological processes underlying the disorder. There are some challenges in psychiatric diagnostics because it is based on the Diagnostic and Statistical Manual of Mental Disorders, and the whole process of diagnosis is based on an interview. And a diagnosis based on observation or an interview can lead to bias as well as heterogeneity. So we believe that it would be very important, or it is very important, to study the underlying biological processes of mental disorders to be able to support diagnosis with hard evidence. One of the potential pathophysiological mechanisms underlying schizophrenia could be an imbalance of cortical excitation and inhibition. And transcranial magnetic stimulation combined with electromyography is a good tool to study these cortical excitation and inhibition in schizophrenia. So the aim of our first study is to study these paradigms. And to help you better understand how these paradigms look like, imagine that there is a magnetic stimulation over the motor cortex, which results in a muscle contraction in the hand. This muscle contraction can be measured with electromyography. Depending on the strength of the stimulation and the time between two stimulation points, we can distinguish between inhibitory and excitatory responses and paradigms. In my first meta-analysis, we are comparing 4 of these paradigms in schizophrenia patients and healthy control group, and we hypothesize that there can be a clear difference between schizophrenia patients and healthy control group. You can see my selection and search protocol here. We ended up with 36 articles in the analysis. Here I'm showing you the results of my first outcome measure, which is short interval intracortical inhibition, SICI. This is an inhibitory TMS-EMG paradigm. SICI is expressing the amount of inhibition, and we studied that in schizophrenia patients and healthy control groups. SICI is a ratio number, and the higher the number, the smaller the inhibition is. So for example, an 80 of SICI means 20% of inhibition. You can see on the forest plot, those articles that are from 0 to left found that there is an increased inhibition, so bigger inhibition in schizophrenia patients compared to healthy controls. Whereas most of the articles that are from 0 to right found that there is a reduced inhibition in schizophrenia patients compared to healthy controls. You can see that my pooled effect size is mean difference because the articles reported means and standard deviations of SICI. You can also see that this result is not very heterogeneous, and there is a strong statistically significant finding that there is a reduced inhibition in schizophrenia patients. We believe that this result is very important clinically as well as statistically. Moving on to my second outcome measure, cortical silent period. This is also a TMS-EMG paradigm of motor cortical inhibition. And we compared that in schizophrenia patients and we studied that in schizophrenia patients and healthy controls as well. Cortical silent period is a length of the inhibitory response. So the numbers you can see on the plot are the length of the silent period in milliseconds. You can see the articles from 0 to left, or in those articles, they found reduced inhibition in schizophrenia patients compared to healthy control, whereas the other side of the from 0 to right, you can see those articles where they found increased inhibition. You can see that this plot is very heterogeneous, and we haven't found a clear statistically and clinically significant difference between the two groups. We did multiple subgroup analysis and exploratory analysis to trying to understand what is the reason for this heterogeneity. However, we didn't find any clear one moderator variable that can explain all the heterogeneity. We think that there can be possible reasons for heterogeneity. One of them is biological variability, measurement bias, and publication bias. We believe that there are some strengths of this meta-analysis. First of that is that this is the only and first meta-analysis that compared all these TMS-EMG paradigms in schizophrenia. We could include a large number of articles in the analysis, which resulted in some strong results in case of psyche. But there are some limitations to this meta-analysis. One of them is that within each article, they use small sample sizes, and there were some heterogeneity that you could see in the case of cortical silent period. This heterogeneity is rooted in experimental conditions and sampling methods. And there were some statistical bias in the case of cortical silent period as well. So as a conclusion, we can say that the results for psyche are robust, and we found a clear difference in psyche between schizophrenia patients and healthy controls. Therefore, we believe that psyche could be possibly used to support diagnosis of schizophrenia in the future. However, cortical silent period is not strong enough to be used as a potential biomarker for schizophrenia. Based on our results, we have some clinical or implications for future results, and we think that it's very important to include more patients and use bigger sample sizes in future results and avoid statistical bias as well as some methodological bias. You can see the progress for my first meta-analysis. We are working on a manuscript at the moment. Moving on to my second project, which is also going to be a meta-analysis. Here, I'm also studying the underlying neurobiological mechanisms of schizophrenia, but with resting state functional MRI. I am going to study thalamocortical resting state functional MRI connectivity in schizophrenia, and it is very important because schizophrenia has been described as a disorder of this connectivity. And resting state functional MRI allows us to study connectivity in the brain. A thalamus plays a very important role in cognition as well as perception. It has a role of a gatekeeper, or it has been described also as a relay station that connects sensory organs to higher level cortical regions, as well as it has some connection directly for higher level cortical regions. It has been described that there could be a deficit or abnormality in thalamocortical connections in schizophrenia. Therefore, we believe that it is important to study that in a further meta-analysis. So our aim to assess functional resting state MRI in thalamocortical connectivity. And the preliminary research supported our aim, and based on the preliminary research, we think that this could be a feasible topic for second meta-analysis. So thank you very much for your attention, and I am looking forward to your questions.
Amazing presentation. My question is for your first project. I would like to ask how specific these outcomes are to schizophrenia.
That is an interesting question. These TMS-EMG protocols have been studied in some neurological disorders, for example, in Alzheimer's disease and Parkinson's disease, but we don't know the specificity to schizophrenia because we only compare them to healthy controls. But it's an interesting question.
Congratulations to your nice talk. May I ask you how frequently this transcranial magnetic stimulation is available for these patients with schizophrenia? Is it a routine or is it underused?
Okay, so transcranial magnetic stimulation usually is used for, or has another use in therapy, and it is currently under research, the effect of transcranial magnetic stimulation therapy in schizophrenia, so it is not very well used. And these kind of paradigms that I talked about, these are also not very widely used so far. But there are some research in this field, so we believe that it was important to study that.
And how about the availability of the electromyography for the psychologist and the psychiatrist? Can they request in enough number? Because I think it is done by the neurologist.
Well, because transcranial magnetic stimulation therapy is available or used, for example, in depression, there are some psychiatry departments where TMS is used. So I think it would be possible to use that and combine that with electromyography as well. They use surface electrodes in these paradigms, not needle electrodes, so the technique is a bit easier, and electromyography can also be used in other psychological studies as well.
Thank you. May I ask a naive question? So whether it is easier to work with patients suffering from schizophrenia or depression?
I don't have an experience with that one because I don't work as a clinician. I focus on research, and I have some experience with data collection or examination with schizophrenia patients, but not much with depressive patients.
Thank you.
My question is that can you somehow measure the results of the stimuli directly in the brain instead of muscle movements?
Yeah, that is a very interesting question because in case of schizophrenia or psychiatric disorders, I would be more interested in other regions of the brain. And there are some techniques that combine TMS with EEG or MRI, which allows us to study other responses in other cortical regions. But these are very novel techniques. There is not much research on this field, so it is not a good topic for meta-analysis, but for future research, it is a very interesting topic, yes.